Okay, so yeah, okay. So let me go back, and this is something where we left off last time, and we were looking at basically microbial ecology and trying to understand, for example, in a very complex ecosystem like a wastewater treatment plant, what are the biochemistries and how do the different sets of microbial communities which are associated with a distinct function, how they are interacting with each other in order to convert your organics which are in the wastewater pipeline into finally methane, okay, if that is the case. And then we also started to look at, for example, the measures of microbial diversity, and this is where we had left off last time. So one of the points which I had told you was that in general, in any healthy ecosystem, we have found that the microbial diversity is much higher. And by that, what we mean is that the number of species which are given in that particular ecosystem are going to be much higher compared to that which is not a healthy ecosystem. And therefore, sometimes microbial diversity can also be taken as an index of monitoring eco-health. All right, so here was an example where we left off last time that on the x-axis here we have 16S ribosomal RNA richness. In other words, if you take, for example, a soil sample from a forest, for example, and then you try to extract the total DNA, and with the universal primers, you try to find out, for instance, how many different type of 16S DNA molecules are amplified. And by virtue of having very powerful electrophoresis techniques, all these fragments of DNA, they could be identical in length, but in terms of their nucleotide composition, even if they are different from each other by one nucleotide or two nucleotide, all of these can be separated on this gradient gels. So the number of such bands on the gel is an indication of how many different type of species are there in the sample. Okay, so is that clear to everybody, that point?
Yes, ma'am.
Okay, so that's what we are trying to plot on the x-axis. All right, so the relationship which you are trying to measure is between the 16S ribosomal RNA richness, and on the y-axis, we have the metagenomic richness. That is, here, the entire DNA is taken and it is cloned, it is also sequenced in order to know what kind of functions are carried out on that particular DNA fragment. Now, unlike in 16S ribosomal RNA, where you are only amplifying the gene which is coding for the 16S ribosomal RNA, in the metagenomic library, you also get a chance to look at what are the functions which these DNA are coding for. What enzymes are they coding for? What is the functional diversity? So on and so forth. So the data which is collected is for example, the inverted, the triangles which are here are the soil samples which are taken from the cold desert. Similarly, the red triangles are the soil samples which are taken from a hot desert, so on and so forth. So all of these symbols are explained here. And what you find is that in this community, for example, if you look at cold desert, they couldn't measure more than 5,000 different types of species. That is the maximum they found. So between 3,500 and between 5,000. Okay, that is the different type of species. And if you look at the function here, for example, okay, you do find that there is a lot of functional variability here. So the species richness may not be there, but there is a functional variability starting from 1200 to 2200 different type of functions are encoded. Okay, then when we go on to, for example, other samples here, again, as the number of, for example, richness increases, you do get to see some kind of an increase in functional diversity. However, there is no specific correlation between the two. Okay, so R is the correlation factor, which is not very high here. But the point here, which is being made here and which needs to be noticed, is that if you look at extreme environments like the cold desert or maybe the hot deserts, all right, the species richness is not as much as it is, for example, in this temperate deciduous forest, or you have the temperate coniferous forest, so on and so forth. So in other words, extremes of conditions do not allow for too much of a microbial diversity to occur. It's only in the temperate areas that you do see species richness here. Okay. That is one point. And number two, this is of course the difference in the alpha diversity. Okay, then there are various ways of putting down this diversity. So there are numerical methods whereby diversity can actually be measured. This is of course only one way, but you can also report your diversity in a microbial population as what is called as an alpha diversity. What does it mean? It means the total number of species which are present in a sample, that defines its alpha diversity. And it is determined using species richness or a diversity index. So either one of these parameters can be used to say what is the alpha diversity. Then what is beta diversity? Let's say you have two different soil samples. One is from a temperate forest, other is from somewhere else. What is the difference between the two? Okay, is called as the beta diversity. Gamma diversity is a sum of both alpha as well as beta diversity, which is the overall diversity in a sample. Okay, so species richness is another parameter which is used. That means it is number of species in a sample. Species evenness, that means let's say there are 100 different species. Okay, one of the species is represented by 25 organisms, but the other species, okay, is only represented by 2. That means there is an unevenness in the species. Okay, but let's say if there are two species, both of them are represented by equal number of microorganisms. You have 25, you also have 25 of the other one. That means this is evenly distributed. That is what we mean by species evenness. Okay, so that is also another way of measuring microbial diversity. So all of these parameters can be used. And we had very briefly touched upon alpha diversity. So here is a very simple picture. So is this clear to everybody, all the terms which we have used?
Yes, ma'am.
All right. So in this picture, you will have a very good idea of how to measure alpha, beta, and gamma diversity if you are provided with some data. Okay. So in site A, for example, we say that the alpha diversity is going to be 3 because it defines the number of species which are present. In this particular sample, you see orange color, there are 2 microbes which are of one particular species. There are 3 microbes which are of another species, and then there are 6 of the third one. So the alpha diversity will be called as 3 here. In this sample, the alpha diversity is again 3. In this sample, the alpha diversity is 2. There are 2 different types of species which are present. When you look at the beta diversity, let's say beta diversity is always a comparison. So here it is between site A and site B. So it is difference. So in this sample, there are 3, out of which 2 are going to be common. Because you see the orange one and the green one are common between A and B. So you say the beta diversity will be now 3, that's what is here, and then 2 are common, so you deduct that, 3 minus 2, plus in this side there are 3, out of which 2 are going to be common. Okay, so therefore, as is written here, that the beta diversity between A and B is equal to 2. And the number comes from the fact that you had 3 here, out of which 2 were common. Similarly, you had 3 here, out of which 2 were common. So that is how 1 plus 1 adds up to 2. So you will say that between A and B, the beta diversity is equal to 2. If you look at B and C, okay, again, there are 3 different species here. You see here 3, but one of them is common, the green one. So you say 3 minus 1. There are 2 species here, out of which 1 is common. So again, you just add them up, and that is how you get the number 3. So in other words, in ecology, okay, a population, the species richness or the diversity, etc., these are different ways by means of which you can define it. And then gamma diversity, as you see, is, how many different species are overall present in all these microbial populations. So we had 3 here, but then we have an additional one here, 4. We have an additional one here, which is shown by this crossed blue, 5. And that is the reason why we say here the gamma diversity of this ecosystem is equal to 5. Okay, so is this point clear now as to how do you really compute alpha, beta, and gamma diversity in a sample? And species richness, etc. How many 16S ribosomal RNA fragment have you counted will give you the number of species, so on and so forth.
Yes, ma'am. Yes.
Okay, all right. So, this was something which I had explained, but, very briefly in a minute, I will review this again. That is, in any microbial ecosystem, all right, as the species richness increases, okay, so species richness is going from some number, let us say 1,000 to 5,000. Then what you see on the y-axis is that the functions are going to increase. By function, what you mean is that the biochemical capacity of that ecosystem, and that is to be expected. As you add more and more microorganisms, or you add more and more biological systems to an ecosystem, the functional diversity is also going to increase. Okay, but then does it have any role to play with the stability of that ecosystem? So in this slide, that is a point which is being made, that as species richness increases, okay, the functional diversity of the ecosystem definitely increases. Okay, but then it kind of plateaus off. So after this point, what you see, as species richness increases, there is no further effect on the functions which are likely to increase in the ecosystem. And such kind of a data has been collected from different kind of ecosystems. All right, so after some point, so you had 1600, then maybe you had 2000, 2400, 2800, so on and so forth. But after some time, in spite of increasing the species richness, the functions which are associated with the ecosystem are going to plateau off. However, it has a different effect on the stability. Okay, with the species richness, the stability starts to increase, although there is no functional gain for the ecosystem. Okay, simply because there is functional redundancy. You have added 15-20 different microorganisms, 15-20 different species, and they also bring in with them the same functions as you had already accumulated in the ecosystem. However, such a system becomes more stable now. Why? Because if there is an extreme climate of pH or temperature, maybe two species are going to disappear. But since the same functions can also be performed by a number of other species, which you have increased in this part, so therefore the stability of the ecosystem is going to be increased as the species richness increases. Okay, so is this point clear?
Yes, ma'am. Can you please explain what does it mean by stability here?
Okay, by stability is that, let's say any kind of an ecosystem, take lake, for example, as an ecosystem. All right. Now, if there is a sudden change in the temperature or sudden change in the nutrient concentration, so there is too much of pollutants which are coming into the system, and it is likely to impact 2 or 3 different microorganisms which could be very sensitive to that particular pollutant. Let's say cadmium is being added to the lake, and there is an industry which is a chrome, which is a painting industry, so it is releasing excess of cadmium now into that lake. Okay, there could be 2 or 3 species which are extremely sensitive to cadmium, therefore they are going to disappear. However, since there is a species richness in that lake, that is, there are other microbes which are able to carry out the same function as the ones which have disappeared. Therefore, they will be able to take over. They will be able to still survive in that ecosystem. Okay, so there will still be life, will still be present in the lake. It is not that the entire life or the entire ecosystem is going to be killed. Why? Simply because there was species richness there.
Yes, ma'am. Understood.
Okay, that's what we mean here. All right. So now here is a picture which is, from that chapter. I had uploaded it yesterday, that by and large, when you talk about microbial community response, and what is extremely important is that at this point of time, why are we trying to understand about these microbial communities? Because eventually, as a process engineer, as an environmental engineer, whenever you try to address an environmental problem, okay, it could be, wastewater treatment was only one example, but there are pollutants which are from mining industry, for example, or solid wastes which are constantly accumulated in the environment, or textile effluents, or leather industry wastes, etc. If you want to provide an environmental friendly solution to these, okay, you will have to deal with microbial communities. There is no single microorganism which is capable of carrying out degradation or remediation of these complex effluents. You will always have to work with the community. So you need to understand, how do microbial communities work with each other? Okay, what are the different members? How do they interact? Do they benefit from each other? Are they going to, compete with each other? Do they help out each other? So that if you have to prepare a consortium of microbes in your own laboratory, as is generally done in these cases, okay, you prepare a microbial consortium yourself, you make a collection of those microbes, you set up a microbial community, you lyophilize the culture and you provide it to a company. Okay, like oil spills, which are in Saudi Arabia, these are the oil producing countries. And whenever they do, whenever oils are taken out or they are rigged out, there is a lot of spill which occurs naturally. And there are companies which will supply these microbes in a pouch, a community in a pouch, including our own TERI, for example, the Tata Energy Research Institute. They are also in a big way taking projects from the Middle East, where they have these oil degrading bugs which they have prepared. They lyophilize, they pack them in these pouches, okay, and they supply. So wherever there is a spill, these microbial communities are dispersed over those oil spills, and they actually eat up all the oil. So the soil health is restored back to the original one. Okay, but such kind of solutions cannot be provided unless you have an understanding of microbial community and what impacts them. That's the reason why we're trying to understand and emphasize so much about it. So is that point clear to everybody?
Yes, ma'am.
Okay. So anybody else in the class? I mean, do you understand? You talk about diversity, you talk about microbial ecology. Where does it actually fit in eventually in the area of environmental biotechnology? So, you always need to look back, you need to make a connection all the time. So now, here is a picture which illustrates, for example, that microbial communities, how do they respond to outside factors? So any disturbance like you see here, which could be a disturbance in the nutrient, or it could be a disturbance in the pH or in the temperature, all right, then the microbial community, either it is going to exhibit resistance to it, that is, there is going to be no change in it, in which what happens that the original community composition is entirely maintained. Okay, so let's say the pH suddenly, alkaline pH comes into that particular lake. However, the original community composition is completely maintained. That means this microbial community was resistant. Okay, or the community composition could be altered. All right, and the community composition is altered temporarily, and eventually it may switch back to the original community composition, in which case the response is noted as being resilient. So temporarily, in order to handle the higher pH influent or higher pH discharge into that lake, okay, some of the members of the community which are pH resistant, could grow much faster until the conditions normalize. But eventually, over a period of time, maybe 15-20 days, then the original community responds back, because it has restored itself, in which case you say it is a very resilient microbial community. Or the community composition is going to be altered, which could be of a permanent nature. So you now have an altered community which functions very, very differently, or altered community that functions very much like original community, because they have acquired some more functions in here. So in general, this is what needs to be kept in mind. This is how the microbial communities are going to respond. Okay, and based on 70 different studies which have been conducted, case studies, okay, the data indicates that the microbial communities are extremely sensitive to following factors. Number one, 60% of the microbial communities which have been investigated in these studies, okay, they are sensitive to increased carbon dioxide. Okay, because sometimes you want to know what are the factors which are going to be most critical. Okay, so what we find from the data, 60% of the microbial communities are extremely sensitive to carbon dioxide, and that is the reason why global warming is such a big problem, because we know it is going to alter carbon dioxide generation. Dissolved carbon dioxide which is present is also going to alter the community composition, and that can change the biochemistry of Earth. 81% 84% of the communities are sensitive to nitrogen, to phosphorus, and to potassium fertilizers. 82% to temperature, 83% to carbon amendments. That is, if you bring in some other kind of a carbon, organic carbon, or you bring in aromatic, aliphatic hydrocarbons are now being pumped in. Other than the normal carbon sources, 83% of the communities are going to display some kind of a response to different kind of soils which have different organic carbon. Okay, and that's the reason why, in other words, out of so many factors which we investigate, when you talk about environmental pollution, what is it that I should be focusing on? There are so many pollutants, this, that. So how do I draw up my priorities? The priorities should be drawn based on this data. Okay, so is that point clear?
Yes, ma'am.
All right. So now, before we leave the topic of microbial ecology, let me just tell you, as an engineer, for example, that your job is to design and operate the engineered processes. Okay, as a process design engineer, what do I learn from microbial ecology and how can I implement it to find solutions? Okay, so the basic questions which you are going to ask as a process engineer is what microorganisms are present in this microbial community so that I can use them to my benefit. I have a project on bioremediation to carry out. So the first thing which I want to know is what microorganisms are normally present. Let's say there is a water body, which you certainly want to investigate first the natural systems before you start to operate a reactor. Okay, so you are going to ask what microorganisms are present. Okay, in other words, what you are trying to find out is the community structure. Enumerate, number them, how many different types are present, abundance, that is, there are 50 different species, but each one of them, how much is it represented? Is there 2 of a species? Is there 50 of a species? Is there 100 of a species? This is what it means. Enumeration is getting the number right. Abundance, how many are present, and spatial arrangement. How are they arranged spatially? What do we mean by that? So when you look at a wastewater treatment plant, there are granules which are formed. Okay, now granules are nothing but these are 3-dimensional structures in which there is a spatial orientation of different microbes, more which are towards the surface of the granule on the outside or on the first layer of the granule. Looking from outside are the ones which are going to be aerobic organisms. As you move deep down into the granule, there are going to be anaerobic microbes. Okay, so in a flock, for example, because generally in a in activated sludge process or in any anaerobic digestion process, flock formation, granule formation is the process is always accompanied by formation of these structures. Okay, so you need to understand, for example, what is the spatial arrangement of these members of the community. Then you want to know what reactions could the microorganisms possibly carry out and what reactions are they actually carrying out. So what are you trying to study is the community function. How are the different microorganisms interacting with each other and the environment? In other words, you want to find out what kind of materials are produced by them, what are they consuming, and what materials are they exchanging between them. Again, to go back to the typical wastewater treatment plant, the community structure. You have the hydrolyzing microbes, and then you have first the aerobic ones. Then there are the fermentative bacteria, which take up sugars. They go to volatile fatty acids. And then you have the anaerobic bugs in these flocks, which are able to make acetic acid and then acetic acid to methane, which is completely an anaerobic process. Okay, so what materials are produced, acids, etc., what is being consumed, what is being exchanged. So carbon dioxide, hydrogen, volatile fatty acids, these are being exchanged. Okay, so when you talk about interactions between the different microorganisms in the community, you are definitely trying to find out what materials, and what are they really exchanging with respect to each other. And only then you are in a position to operate such kind of a reactor. Okay, so as applied microbial ecologist, so you can say an engineer who works on plants like these is basically an applied microbial ecologist. Okay, so they create a system which is a reactor in which the right kind of microorganisms are present. That is, you have made the community structure, you have understood. These are accumulated to quantities which are sufficient enough to complete a desired task, which is the community function, and they work together to perform their tasks stably over a period of time. That is degradation of COD or degradation of BOD, finally lowering of the BOD, lowering of the CODs, and that is the end task which they are supposed to perform. Okay, and so you have learned about all of this. For this, nutrients are supplied to the desired microorganism. You try to retain those bunch of microorganisms, allow them to make adequate contact with the material to be treated, which is in this case waste material, which is a solid material. Okay, you have to make sure that there is an adequate contact between the microorganism and the wastes. So that is the job which you are going to do, and you will draw out from the basic knowledge of microbial ecology, which poses these questions which you are going to understand and eventually apply them to such kind of a reactor. Okay, so is that point clear to everybody?
Yes, ma'am.
Yes, ma'am. Okay, anybody else in the class? So anybody else, please? So, I would very much like that, because, sometimes it's important to make a connection also on very, very different disciplines. When you're trying to bring them together, you need to make a connection. So here you are drawing out from the principles of microbial ecology and then applying them for, design and for operating a reactor. So anybody else has a question? So the class, are you listening? I don't see, except for one or two people. Uh, Deepak Kumar, are you here?
Yes, ma'am.
Radhika? So Deepak, can you now tell me, like, we talked about why do we choose 16S ribosomal RNA for determining, the composition of the community? Can you answer that question?
Because, ma'am, ribosome is necessary for encoding of the protein, and protein are the building blocks of the life. So it is ensured that ribosome will be present in every organism. So we sequence the gene which encode for the ribosome. So on an basis of that, we can define that because every organism will have a different ribosome according to its variations in its 16S ribosomal RNA, and on basis of these variation we can decide how many number of different microorganisms are present in the community.
Certainly, very good, very good. Vivek, would you like to say something about the community structure which you have learned in for the wastewater treatment? Okay, what are the different reactions which are going on? Vivek, are you there? Vivek, Sarah? I'm afraid everybody has to make an effort because these are very difficult times. And unless students take the responsibility of being in the class and learning the subject, you will just pass through the course without learning anything and also get a bad grade in the end. Sumit Anuragi, can you answer that question? What are the different four different phases which we discussed in the anaerobic digestion process? Sumit Anuragi? No response. So, do you just log in and go back to sleep, or what do you do? Arish Khan? Yes, Sumit, yeah, you have just sent a note. You can unmute yourself and you can answer the question, Sumit. Why is there a mic problem? Just give it in writing, on the chat box, just the question. Please send an answer to this. And Sumit, if you have a mic problem, just please send an answer to the question in the chat box. Jatavat, are you there? Arish Khan. So, as it is, there are just 10, 14 people and, attendance is mandatory. So what is your, eventually, how do you think you are going to pass the course? There is no attendance. You are not participating in the lectures. You are not alert during the lectures. So how do you think you will just copy in the end? Everybody will get a different question paper. And, then I will have to conduct major exam in the form of a viva. Gopika Arora. So I don't know who are you cheating really? Just yourself. Yeah, Gopika, you have raised your hand. What is the problem, Gopika? Can you unmute yourself? Or can you send something on the chat box? So, very poor response. I don't know what is the problem. Mic isn't working. Why not? You are in a class. Look, all these things you're supposed to be figuring out before you start the semester, that your camera is working, your mic is working, and eventually everybody cuts a sorry figure, and it's embarrassing for the student, it's embarrassing for the teacher to be teaching in a class, where you're not even sure if students are listening. So मतलब हर कोई यहाँ पे मौन बैठा हुआ है. रत्नेश, are you there?
Yes, ma'am.
So I'm afraid, people, you will really have to end up in a misery if you don't straighten up your act, and if you don't, because these are not courses which you can just learn overnight. One day before your exam, you open and suddenly you become master of the course. It's not going to happen. So, so in the last lecture, in the last slide, overall, we try to sum up. And so the kind of, all the questions which we had raised here, what microorganisms are present, etc. And generally these solutions are, categorized in the following categories. That is, you are going to be able to, you will have to select the organisms, because if you are trying to build up the right kind of a community structure, you will have to select the organisms, or you will have to manipulate your environment so that the best microbes are selected, or you will have to select conditions which lead to aggregation. Why? Because, as I said, that flock formation or granule formation is a very characteristic feature which is associated with these, with the community development, or with the operation of the community. So experimental conditions, operating conditions will have to be altered, which lead to aggregation of microbes. You also have to be in a position to control exchange of materials. So how much of substrate you are going to feed in, the genetic information, what conditions should be kept in the bioreactor in order to control the exchange of materials is something which you will do. Adaptation or acclimatization is another process where as soon as you take an inoculum, sometimes it doesn't work right away, but rather it takes time to adapt to those conditions. Okay, so these are some of the solutions which are available to you whenever you want to work as an applied microbial ecologist and to working of such a bioreactor. So finally, before we leave this topic, we also in the literature, we see terms like microbial mega communities. So whatever we looked at right now, they do not really classify. So the microbial communities which are present, let us say in a wastewater treatment plant, cannot be called as a mega community per se. Okay. And mega communities have a very different kind of an understanding to them. And these are basically structures which are very, very unique and which are composed of a limited number of microbes. And yet these are very, very specific. Okay. In these communities, these are basically bacteria which like to adhere to surfaces. Okay. And the surface associated microbial communities. Now, for example, if there is an implant, okay, now when people undergo surgery and other things, they do have implants in the body. For example, a catheter can be added to a patient for 20-25 days or whatever. So that is something which goes into the body. Or there are metals or rods which are inserted in order to rectify the function of a bone, or stents are introduced into the body for people who have heart problems, etc. But over a period of time, surprisingly, there are microbes which start to grow on these. And these are very specialized communities. And you can imagine how much danger they can pose. Okay, these are generally surface associated microbial communities, and they also have very high concentration of the microbes. Then those microbial communities which are suspended in water column. So if you look at something like a biofilm, okay, you must have noticed that in a flask sometimes when you are growing microbes for an extended period of time, after 60 hours or after 70 hours or something, you will see that there is a layer of a slimy material which is around the glass. Okay, that can be called as a microbial biofilm. Okay, they contain a large number of these colony forming units, which are nothing but microbes. So something like a biofilm, which is a mega community, could contain 10 to the 8 to 10 to the 10 colony forming units per centimeter square. Rhizosphere, which is a very specialized area surrounding the roots of a plant, can also contain so many CFU per gram soil. Okay. So here is a picture just of a biofilm. Okay. And as I explained, many of these biofilms, they can grow on medical implants and other things which are inserted into the human body. Okay, so these are like the solid surfaces on which microbial communities will start to build. And they are very dangerous because, they become resistant over a period of time. They become resistant to antibiotics and they start to secrete their own materials, which can be very, very toxic. And similarly, here we have the rhizosphere. So here is a plant and these are the roots. Okay, so the soil, which is now associated, the one which is in the immediate neighborhood of this, not attached to the roots, but the soil which is just next to these roots is called as the rhizosphere. Okay, and that is a very different kind of a composition. And these are also called as mega communities. Today, there is a lot of interest, either a medical interest or agricultural interest in these kind of communities. Okay, so with this, we end our discussion on microbial communities. So is this, yeah.
What can we consider as a colony forming unit in case of rhizosphere? And again, what is colony forming unit? In a colony forming unit, there can be one or a few bacteria, and there can be many also.
Absolutely, absolutely. So for example, if you take 1 gram of soil, okay, like when you see how the numbers are going to be represented, okay, let us say you have a soil or from the biofilm. Okay, there is a biofilm which is growing inside of a flask, and you just remove you cut out a centimeter square patch of that. Okay, and now you put it in water and eventually you just put it on LB medium. And now you see how many different colonies are going to be formed. So what you see in 1 centimeter square, Okay, everything, for example, in 1 centimeter square can be dipped in water. It can be all extracted into an aqueous solution. You can make dilutions of that, and then you can just put it on LB plate. Count the number of colonies which have come up. Okay, so this way, so let's say 1 centimeter square of biofilm is suspended in 1 ml of water. So all the microbes are now going to be discharged into that 1 ml, which actually comes from 1 centimeter square of the biofilm. Of that 1 ml, you can do serial dilutions, because if you plate that entire 1 ml, you can't count 10 to the 8, 10 to the 10 colonies in a plate. It's just too crowded. If you were to do that, you will not be able to count individual colonies. So what you can do, out of that 1 ml, you can make 1 to 50 dilution, make 1 to 100 dilution, 1 to 200 dilution, and then you plate out that on an agar plate which contains LB. And then you see what is the number of colonies. So if you perform that experiment, you will see that the number of colony, so that's what it is, the number of colonies which you are able to get, okay, out of 1 centimeter square of biofilm will be at least 10 to the 8 to 10 to the 10 colonies. Similarly, if you weigh out 1 gram of soil from rhizosphere, and then you find out what are the number of microorganisms, this is the count which you are going to be able to come up to. And that is how we can put a number to it, that there are so many different. Of course, then you will determine that, are they same species? How many different species are there? What is the abundance of each one of them? So on and so forth. Okay. But if you look at the total number of colonies, that is what this number refers to. So at this point, I will only say that, when you talk about microbial communities, certainly there is a lot of interest in these mega communities because they could have medical implications, or in agricultural practices, these are going to be very, very important. Okay.
Ma'am, I had a doubt in the slide where we discussed the effect of disturbance on microbial community.
Okay, I'll just quickly go back. This one?
Yes, ma'am.
Mm-hmm.
The thing is that if it is resistance, the community and the function will not change, and if it is not resistance, in that case, in the third outcome, the community changed and the functions are also changed.
Yes,
But in
yes.
the in second case, the community changed, but the function is still retained. It can perform that function, right?
Exactly, exactly. Because see what happens when you say altered microbial community, first of all, it has to do with the functional redundancy. Okay, because as we saw in any stable ecosystem, okay, there are different microbes. There could be 10 species which are capable of breaking the aromatic hydrocarbons. Okay, so if it is impacted by a factor which is going to affect two of the species, but rest of the eight, they are still there, they may not have been present in very large numbers in the earlier stages. So what happens is that the functional redundancy happens.
Ma'am, you were not audible.
Ma'am, you're not audible.
Ma'am, you are not audible.
There was an issue with the internet connectivity, so I hope you can hear me now.
Yes, ma'am.
Yes, we can.
Yes, ma'am.
Okay, so there was a question which somebody had asked. So will you please continue with that question?
Ma'am, I wanted to ask that if the community is changed, how the functional redundancy is still maintained in the.
Okay, yeah. So, in that figure, I'll have to upload that again now. So just please give me a minute. Okay, so let me go back to that slide and so can you see the slide now?
Yes, ma'am.
Yes.
Okay, all right. So when we say that the community composition is altered, all right, so it doesn't mean necessarily acquiring new communities from outside. All right, there are microbes which are capable of carrying out identical functions. That is, there is a functional redundancy in the community. But then, let's say there are 10 different species, as I was giving you the example last time, that and all the 10 species are capable of degrading aromatic hydrocarbons. However, two of them are very, very sensitive to the pH, and their optimum pH is so much. The other 8 members of the community, are not able to operate as effectively at that particular pH, but their law of tolerance, is much more. So if there is a sudden change in the pH, the two earlier microbial community are going to completely vanish, but the other two, because the conditions are favoring them now, will be able to multiply and they will overtake. As a result of which, there is an altered community. Okay, so not that new microorganisms, it could just be a gene function. Sometimes there is a change in one particular gene function, and the resulting mutant of that is a little bit better. But still, it will classify that now the community composition has changed. Because in that composition, it is not only the 16S ribosomal RNA, but it is also, for example, how many of each one of them are also present at any given point of time. So there is an altered community now, or it could acquire genes laterally. There could be Pseudomonas in the soil, and through transformation, it picks up some genes for degradation of aromatic hydrocarbons. So it becomes now like a recombinant culture. So the community structure does change. However, it functions like the original community.
Yes, ma'am, got it.
Okay.
Ma'am, I had another question that you said that if there is a sudden change in pH, so is there any parameter to measure that how much fast the change is allowed so that a particular kind of bacteria can survive? Like it is there some kind of parameter defined for every organism?
You
Like
see, first of all, these experiments can be carried out in the laboratory. Okay, like for example, the soils can be taken from a particular area, and or you could do it in the field experiment over a large piece of land. There is some microbial community which you are trying to study, and in that experimental phase, you can introduce, for example, a pH change, and then after 7 days, after 15, 20 days or whatever, you can change what is the microbial community response. So a lot of people have generated data like that. Experiments can be conducted and these questions can be answered, but it would certainly depend from what area you are dealing with. What kind of, where exactly is the soil sample which is coming from. So there is no universal rule which can be applied. Now, for example, in a temperate forest, one of the slides which we saw earlier, that in that kind of an area where microbial diversity is already very high compared to what you see in the cold desert, if you bring about a change in the pH, even very radically, you may see that the community structure doesn't change so much. But if you have something like a desert community and you have a soil sample from there, you change the pH exactly the same way that you did in the other sample, but you will see that sooner or later that community will not change and eventually nothing will grow in that because there was no diversity to begin with.
Okay, ma'am.
All right. So such kind of experiments have been performed and there is no universal law. But yeah, there are case studies from where we can learn as to what happens in nature. Okay, so with this, I close my discussion on microbial ecology, and now I'm going to begin the next topic, which is called as ecotoxicology. All right, and ecotoxicology is a subject we are going to see is extremely important, where you are trying to determine the toxic nature, toxicology of a given pollutant towards ecosystems. Okay, the only difference is that normally whenever you talk about a pollutant or a contaminant, you will say what effect does it have on humans, or what effect does it have on a given microbe, on an E. coli, or let's say on the birds or whatever, like many examples which we have discussed in the lectures so far. So that is where a single pesticide or a herbicide, for example, is extremely toxic to, let us say, the bald eagle or to some other this thing. Okay, but when you talk about ecotoxicology, you try to evaluate how does the toxic nature of a given compound affect the entire ecosystem. That is what the discipline of ecotoxicology is. And in order to understand this, there are tests which have been devised. Okay, and that's extremely important for you to know, because in your hand you could be dealing with a new kind of a compound and you want to evaluate how toxic is it really to the ecosystem. So that's what we learn in the subject of ecotoxicology.
Ma'am, I had a question from last slide.
Okay.
About rhizosphere. So, ma'am, what can we do about the rhizosphere? How can we engineer that? Like, what's the how can that be like ensured for more benefits?
Uh, when you try to study, for example, so in a healthy field, as I said, that you have, especially with the legumes, that is our, dal and things like that. And if you examine their roots, they have root nodules. By nodules, what we mean that they, it's like a bulbous structure which is associated with the root, and within that
Yes,
are resident microbes which are actually nitrogen fixing. Okay. Now, in
yes,
these rhizosphere, there is a unique population of cells. Maybe I'll just very quickly go back to that slide and spend. Although I did close that. Okay, so can you see this slide here?
I'm not. I cannot.
I see. Do I need to go back and okay, so can you see this?
Uh, Not yet, but maybe it is visible.
Are you able to see the slide now?
Yes, ma'am.
Yes, ma'am, we can see and hear.
Okay, very quickly. So let's try to finish. You had a question in this. What was the question?
Ma'am, my question was like, why we want to engineer rhizospheres, and what's the purpose of that, and how can we engineer?
See, because number one is to reduce your dependence on artificial fertilizers. For example, nitrogen is always supplied in the form of urea. Okay, now the question is that once you add these chemical compounds like urea, etc., uh, there is a downfall of microbial diversity in that particular soil where all the fertilizers are added, and plus the quality of the soil also goes low. So the question is that if the objective is to be able to supply the nitrogen only, then why not have a close association of the plants with a special kind of a soil which already harbors the nitrogen fixing bacteria. So when you say that rhizosphere engineering you want to do, that means you would like to enrich in the rhizosphere those kind of microbes which are able to fix nitrogen, or which are able to release other kind of end products which are going to be beneficial to the plants, so that they are no longer dependent on external phosphate addition or external nitrogen addition, because there are phosphate solubilizing bacteria also. So when you want to do, because phosphates have to be supplied, it's a major element. Nitrogen has to be supplied to the crop. Okay, so nitrogen you have in the form of urea, phosphorus is supplied in the form of organophosphates. Okay, so
Yes,
all of
ma'am.
these are externally added to the soil in order to provide them to the plant. But what if there are phosphate solubilizing bacteria which are able to, the inorganic sources of phosphates which are already available in the soil, these bacteria can mobilize the phosphate from those sources. Then you don't have to
Ma'am,
supply these from outside. Okay.
in a rhizosphere, in the roots of these plants,
Yeah.
these plants which carry rhizobium, from where these bacteria comes? Like, these come through the seeds, or they are already present in
No,
the environment? They grow. They come when they come in contact with that plant root. They start growing.
absolutely. They are already in the environment. Even if there is, see today, of course, on the agricultural land, there is crop rotation. So between two major crops like wheat and rice, for example, they try to put another crop, which is generally either peanuts, because they are all legumes. You know, they have these seed containing peas, or your pigeon pea, which is our arhar, or which is our, for example, your peanuts, etc. So in their, no, in their roots, there are naturally these bacteria which are capable of nitrogen fixation, so that now the soil which could be depleted of all of these sources, their nitrogen content, etc., is going to be restored by plantation of these crops. So they naturally bind within the soil. These microbes are already present. They may have gone away because you have supplied urea or organophosphate. But when you bring back these other crops, these bacteria will come back in the soil. Okay, sometimes you can certainly add them from outside as an inoculum also. Okay, but by and
Yes,
large,
ma'am.
these microorganisms are already, because this has been going on for years. That the presence of these microbes has already been noticed, so on and so forth.
Yes,
So,
ma'am. Perhaps nowadays people are giving this inoculum, like in the for in blocks. I know that they were providing some kinds of pouch in for mixing this seed with these seeds,
absolutely,
and
absolutely.
yeah.
And they are, maybe they are called as PGPRs, like it says here. These are the growth promoting rhizobia, etc., which are provided. And in this picture, what you see on the right is, for example, if you look at the microbial community composition. So if you see the community composition in the free soil, that is the soil which is, let's say, the soil which is a little bit far away from the roots. The composition is going to be like this. If you look at the composition, exactly the soil which is right, attached to the root, you will see something like this. But in the rhizosphere, which is the soil loosely bound to the root, Okay, that is the rhizosphere. The community composition you see is quite different here. You have more of these, you have less of the green ones here. So the community, and there could be some very unique microorganisms which are present in these rhizosphere. And their composition is different, so on and so forth. Okay, and the idea is that once you have an understanding of what exactly are they doing, what functions are they doing, you can make such kind of a microbial community artificially. You know, all the members of this community can be cultivated in a laboratory. You can mix them in the ideal proportion. You can lyophilize them, make small pouches out, grow them in a bioreactor on large scale, and put them in a sack and then just supply it to the fertilizer, because their dependence on external fertilizers will also reduce if these growth promoting bacteria are given there. First of all, they will be cheaper, they will be healthier for the soil. So that is a practice, and of course, by and large, all the lime, the fertilizers, the organic matter, it will also reduce our dependence on these external things. That is the whole idea. Okay, so I hope that point is clear to everybody.
Yes, ma'am.
Yes, ma'am.
So now let's switch to our ecotoxicology, which I had started to talk about. Okay. So I hope the first slide is visible to you on ecotoxicology. So I had just explained as to what is the meaning, and here is a little bit of a descriptive this thing, that microbial ecology actually or microbial ecotoxicology actually talks about the relationship of the pollutants to a species community, and what is the relationship between the presence of a given pollutant and an ecological system. That is the field of microbial ecotoxicology. Okay, so now the question is that whenever you want to understand the subject of ecotoxicology, first of all, you have to understand and determine what is the level of a given toxin in the ecosystem, and then to classify these. That is, whether you will say that this particular compound is very, very toxic. And so there are different ways of classifying those also. Is this a chronic toxicant? Is it an acute? Is it very severe? So on and so forth. So there are terminologies which are used to define the status of a given toxin in the environment. Okay. So the first question is, how do you determine the level of toxin in the environment? And what do you call any, toxic compound? Or yeah, somebody had a question?
Yeah, ma'am. First, we need to define toxins. What are toxins here? Like,
Exactly.
there can be many.
So toxin is actually a compound, okay, which vis-a-vis a control system. If there is an effect on the growth of a microbial population by the presence of that compound, then it will be called as a toxin. So in that way, okay, it is no different from what we call as a pollutant. The definitions which we have used of a pollutant are exactly the same. That is, it is some kind of a compound which is generally man-made, which is present in the environment. Okay, and as a result of which some biological function is going to be compromised. That can be called as a toxin.
Yes, ma'am. Okay.
Okay, so when you say determine the level of a toxin in the environment, that is, you want to quantitate it, you want to measure it, okay, then the best way would be to quantitate it. Okay, so let's say there is a compound A, what I call as pseudoephedrine, and I want to know what is the level of toxin. So maybe I can take that sample and I can run it through the HPLC, and then I can quantitate. I will know, okay, there is 10 microgram per gram of soil which I have detected. Okay. However, there are two major problems to quantifying by this method. Number one, that this kind of an analysis can also be performed on a given number of compounds. which are prescribed by the environmental legislation. So within the environmental preview, there are 10 compounds which have been already called as a toxin. What if I have a new compound for which the environmental legislation has not really defined that compound, and yet it affects the growth of the microorganism? Okay, so therefore so it eliminates, in other words, those compounds which have not been identified as yet. Okay, so that is something which should be understood, that first of all, it clearly doesn't allow any new kind of a contaminant to be identified if you just were to take it and measure its concentration by HPLC, because all that is limited to compounds which are already in the catalog. So you have a chance of missing out something new. So that is a problem in quantitation. And number two is interaction between the contaminant and its bioavailability. Supposing there is a toxin which is already present in the environment, however, it is not taken up by a biological system, then it cannot be called as a toxin, because it could just build up in the environment. As long as the microorganisms are not taking up, or members of the community are not taking up, so they are not going to be impacted by it, so therefore it cannot be called as a toxin. Okay, so that's what we're trying to say here. All right, so what do you really do then when you identify, or when you think that there is a new kind of a toxic material? Then what we do is what is called as a dose response curve. You determine a dose response curve to a standard microorganism or to a standard biological system. Okay, so whenever we study the subject of ecotoxicology, what you are actually trying to do is to study the impact of that particular toxin on some member of an ecological community, and you are trying to look at the dose response curve. Okay, so what do we mean by a dose response curve? That is shown here. Okay, so in this particular slide, on the x-axis, what we have is that as the concentration of that particular toxin increases, so the units which are given here is the milligram of that toxin per kilogram of body weight. Let us say you are trying to measure it for mammals. You want to extrapolate and know whether this particular toxin is going to be very toxic to a mammalian system. You are going to choose rat as an experimental animal. Okay, so you will have maybe 10 rats, all of them will be roughly approximately of the same body weight. Okay, because whenever these treatments are given, the dosage is always defined as milligrams. So 5 milligram of that toxin per kilogram of that body weight. Is this point clear to everybody?
Ma'am, like why we are defining per kilogram of body weight?
Because people, they respond very differently depending on their weights. You will also, sometimes you must have noticed, when you go to a medical doctor, and if they have to give you an antibiotic or something, they will always ask you, for a child, they will ask them to get on the scale and look at what is their body weight. If an adult gets on the scale, they will look at their body weight, and then only whether they have to be prescribed a 500 milligram dose or 750 milligram or 1 gram dose is based on the body weight.
Yes, ma'am, understood.
Okay, so, and the same thing happens because you are dealing with a mammal here. So the dose is going to be milligram per kilogram of body weight. So on the x-axis, you are going to increase that. And on the y-axis, in a typical dose response curve, you are looking at the response. By response, it could be either a change in the color of that, or when it is, for example, rat as an experimental animal, you could look at how many of them are dying because of this toxin. That could be the response. Okay, so depending on what kind of a system we are choosing, there are also standard practices which will tell you what response is going to be monitored. Okay, in this case, we are dealing with rats, so the response is going to be how many are going to survive. Okay, so as you see in this dose response curve, that if you have, let us say, 5 milligram per kilogram body weight, you see 0%. So out of 10 rats you started out with, none of them died. Okay, so the response remains 0. You increase the concentration to 5, again the number is 0. You increase the concentration to 10, again the response is 0. But as soon as you increase the concentration now to a certain level, you see now 20% of the rats have died. Okay, if you increase the dosage to, let's say, 22.5 milligram, 50% of the rats, that is 10 rats have died, 5 rats have died out of 10 which you started out with. So this response is the distribution of, is actually a, it's a distribution within that population, percent response of that population. Okay, so that is what is called as a dose response curve. Okay, because these are natural responses, not all of them are going to die, because physiologically also, although like the kilogram body weight is exactly the same, the age of the rat is also kept the same, but their genetic composition in a population is always different. So they are going to respond very differently to the same dose. And in order to arrive at the correct picture, you need to monitor what is the dose response curve. That is, what is the probability that if I give 22.5 milligram of that toxin per kilogram body weight, if a person is or if a population is exposed, because remember, you are talking about ecosystems here. You're not talking about individuals here. Okay, so what percentage of that population is going to respond? And that is what you plot on the y-axis. So is this point clear? And eventually, you go on increasing until at a certain dose, you see 100%, all the 10 rats which you started out with the experimental sample, all of them have died. So a response like that is called as a dose response curve in ecotoxicology. Now, is that clear to everybody?
Yes, ma'am.
Okay. You could do it with exactly, as I will show you as we go along, when we talk about these tests which are normally performed. Okay, you could do these tests on rats, you could do them on the primary producers, which are phytoplanktons, you could do it on the primary consumer, etc., so on and so forth. Okay, so there you have a population. Let's say you start out with 100 algae, cyanobacteria, and you expose them to a certain toxin level. And after a certain time, whatever, depending on the dose, you decide that after 24 hours, I'm going to see how many of them have died. Okay, so this way you are going to monitor the response of a particular, either it could be a producer like an algae, it could be a primary consumer, it could be a secondary consumer, it could be a tertiary consumer in a population to that particular toxin. That is how the toxicology data is derived, by looking at the dose response curve. Is that clear?
Yes, ma'am. So we have different kinds of various other different dose response curves for all different organisms, right?
Exactly.
And
And
based on that, like, what will be determined about an ecosystem?
Right. So in an ecosystem, you have the primary producers, you have the, green algae, you have the photosynthetic bacteria, and then you have the primary consumers, which could be protozoa, which could be insect. Then you have the secondary consumer, which could be fish. Then there is the tertiary consumer, which could be like an animal, for example. So anytime when you want to define how toxic is a compound, you will see that the standard tests which are available today, they evaluate how that toxin is going to affect all trophic levels. At primary producer level, so there will be a standard test, where some standard photosynthetic bacterium is taken. So when you order these, this thing, you have to order and you have to work only with that photosynthetic algae. Or you have to work with that insect only, so that worldwide over, there is a standardization of tests. I say that, look, I have found that this particular toxin which is released into the environment because of this biochemical activity or human activity, it is very, very toxic. Okay, they will say, well, what is it toxic to? At what level? So there are some standard tests, in other words, which have been devised globally, and everybody follows those tests in order to determine the toxic level of a given compound. And determining these dose response curves also allows you to, for example, define terms like, DNEL or NOAEL, and I will explain as to what these are. Extremely important in our terminology. Whenever you talk about environmental biotechnology, these terms are used very frequently. Okay, first of all, let me explain what is LD50 or what is LC50. LD is the lethal dose, or LC is the lethal concentration. But basically they are the same. So it is that quantity, that concentration in milligram per liter, or that dose in milligram per kilogram, which causes 50% death of experimental animals. Is that clear what we mean by LD50? You see, so it is that dose which causes 50% response. So if I started out with 10 rats, okay, and if at this particular concentration, 5 of them died in 5 different experiments which I carried out, then I will say that the lethal dose, or the LD50 value for that particular toxin, is 22.5 milligram per kilogram body weight. Okay, and this way, obviously, the lower the LC50 value is going to be, the more toxic that compound is going to be. That means if I'm going to shift this value here at a lower concentration, I'm going to be able to kill 50% of that. That means that particular compound is certainly more toxic. So LC50 value therefore defines you to compare across different toxins how one toxin is going to be worse off, or how is it a little better compared to anything else. You simply compare them in terms of LC50 values. Okay, so is that point clear as to what we mean by LC50?
Yes, ma'am.
Okay, then there are terms like NOAEL, okay, which is defined as no observed adverse effect level. Okay, because it's a statistical distribution, and from the plot the values can be actually derived. Okay, so no observed adverse effect level is, you will see, it is the highest exposure level at which there is no biologically significant increase in the frequency over the control group. That is called as NOAEL. Okay, so at this particular level, there is no increase in the response compared to that in the control, and that value can be derived only from this particular plot. Similarly, what is LOAEL? It is the lowest observed adverse effect level, or it is the lowest exposure level at which there is biologically a significant increase in the frequency or the severity of the adverse effect between the control population and the experimental population. Okay, and derived no effect level. So below this concentration there is, and then it also allows you to derive a level like DNEL, and eventually a dose response curve is used to derive, for example, the DNEL, which is actually the threshold safety level. Okay, then you will say that for this particular toxin, the person should not be exposed to a level which is more than 3 milligram per liter. So in any occupational hazard, in any, let's say there are miners who are working in a coal factory, or there are some other miners who are working and they are exposed to so much of asbestos. Okay, so I need to define what is the minimum level to which they can be exposed, which can be considered as a safe level. Okay, so a dose response curve will allow you to build up this DNEL, which is actually, this is derived from the plot, no effect level. Okay, so we can assume that at this particular level, there is going to be no effect at all. And that is what allows you to set up the threshold levels for any toxin. So is that point clear?
Yes, ma'am. Yes, ma'am.
All right, and such kind of dose response curves, the entire ecotoxicology, or the entire, whenever you talk about safety guidelines in any company, they are all looking at basically the data which is derived for those particular compounds, okay, in the dose response curve. And that is how they are going to set up their limits and the targets. Okay, so that's it. So let me stop here, and we have only 3 minutes left now. And so we will come back and we will resume. The lecture. So if there are any questions, you can also post them. exposed them